is Valeria Chichagova and I'm an Innovation Manager at Newcells Biotech. I'd like to present a poster titled Human-Induced Pluripotent Stem Cell derived Retinal Model containing microglial cells as a platform for toxicology studies. Microglia are the primary tissue-resident immune cells in the retina. They are essential for normal development by regulating neuronal survival, and in an adult retina they maintain synaptic structure and function. Under pathological conditions, microglia mediate adaptive responses within the retina. However, they can also trigger neurodegeneration, intensifying the effect of the disease. Retinal organoids derived from hiPSCs provide a human physiologically relevant platform for studying retinal development, disease modeling, and compound screening. Due to their different developmental origins, current protocols for generation of retinal organoids do not give rise to microglial cells. Therefore, to enhance our retinal model, we incorporated hiPSC-derived microglia into retinal organoids and tested their retinal and vasal capacity and functional in response to agents causing retinal degeneration. In order to achieve this, we differentiated control human hiPSCs to microglia. In parallel, we generated retinal organoids in a 96-well plate format to allow large-scale production. At an optimal time point of organoid maturation, we co-cultured hiPSC-derived microglial cells and assessed their ability to integrate. We also tested the response of retinal organoids containing microglia to LPS and two known retina toxins, including moxifloxacin and chloroquine. As part of the characterization of microglial cells, we studied the appearance of progenitor cells using flow cytometry and showed that there was a steady increase in the number of cells positive for CD14 and CX3CR1 over time. We were also able to show that microglial cells displayed typical morphology and were positive for microglial marker EBA1. We also showed that microglial cells upregulated genes associated with mature microglia in a comparable manner to primary microglial cells. We also tested the ability to phagocytose fluorescent beads and showed that the majority of cells were able to do that. In addition, we also tested the ability to respond to endotoxin challenge by looking at the production of 30 human cytokines and showed that their levels were upregulated in response to LPS. And next, we wanted to see whether incorporating microglia enhanced synthetic retina model. Firstly, we used immunofluorescence to confirm that retinal organoids and the co-culture model both contained photoreceptors as shown by the expression of a pan-photoreceptor marker recoverin, and also they contained retinal ganglion cells as shown by an expression of retinal ganglion cell marker SNCG. We also confirmed successful integration of microglial cells by studying organoids using electron microscopy. Firstly, we showed that organoids that contain microglial cells also contain outer segments connecting cilium in a limiting membrane, but also we could see microglial cells integrated into the organoids. Then we also wanted to see whether retinal organoids and microglial co-cultures were able to respond to stimulants, including LPS, chloroquine, and moxifloxacin after 24-hour treatment. We showed that LPS and/or chloroquine treatments resulted in increased production of pro-inflammatory markers IP10, MIP1 alpha, MIP1 beta, and IL-6. Chloroquine treatment resulted in increased secretion of chemotactic factors MCP4 and MDC. Moxifloxacin did not produce production of these cytokines, which is in line with its predicted anti-inflammatory effect within the retina. So to conclude, we were able to derive functional microglial cells from human hiPSCs. Using our newly developed co-culture method, we generated an improved hiPSC-derived synthetic retina model which contains microglial cells and showed their capacity to integrate into retinal organoids. Treating human retinal organoids containing microglia with LPS and chloroquine resulted in increased secretion of pro-inflammatory and chemotactic factors. As predicted, treatment with moxifloxacin did not induce an increase in cytokine release. Thank you.